All right, let's dive into one of the biggest, most fascinating secrets in all of biology, gene regulation. We're going to open up the playbooks that explains how your body builds itself, literally moment by moment.
So think about this for a second. You've got trillions of cells, and almost every single one of them has the exact same DNA blueprint, the exact same one. But a heart cell is completely different from a brain cell, which is different from a skin cell. So how is that possible? Well, the answer isn't in the DNA letters themselves, but in how each cell decides to read those instructions.
And it all starts with one simple idea: access. Imagine your DNA is this gigantic library of instruction manuals. A heart cell doesn't need to read the manual for how to be a neuron, right? It only needs the heart-building instructions. So the first layer of control is all about making the right books available and keeping all the others locked away on a high shelf. The science behind all this is called epigenetics. That prefix epi literally means on top of. So we're not talking about changing the DNA code itself. Instead, these are chemical tags placed on top of the DNA. You can think of them like little sticky notes that tell the cell's machinery, hey, read this part, or nope, skip this one. So how does a cell physically do this? Well, your DNA isn't just a loose strand floating around. It's incredibly long, so it's spooled up really tightly around these proteins called histones, kind of like thread on a spool. And the key is in these little tails that stick out from the histones. By adding chemical tags to those tails, the cell can either loosen the thread to make a gene readable or wind it up super tight to hide it away completely.
Now, another really powerful tool in the kit is something called DNA methylation. This is where the cell attaches a tiny molecule, a methyl group, right onto the DNA itself. And when these methyl tags are there, they act like a physical roadblock. The machinery that reads genes just can't get past them. It is a very, very direct off switch.
And what's so cool about this is that it's not permanent. It's a totally dynamic system. The cell has this whole toolkit to work with. There are writers, which are enzymes that add the tags. You've got erasers, enzymes that take them off. And then you have readers, which are proteins that see these tags and then carry out the order, whether that's to turn a gene on or shut it down. This is how cells can adapt and change over time.
Okay, so that's level 1, controlling access to the DNA blueprint itself. But what happens if the instructions for a gene have already been copied? Well, the cell has a whole other layer of control for that. It's time for us to meet the RNA gatekeepers.
And this is a really important point. A gene gets copied into something called messenger RNA, or mRNA. That mRNA is like a temporary photocopy of the instructions, and it's on its way to the cell's factory to get turned into a protein. But the cell has ways to intercept that message before it ever gets delivered. One of the star players here is a tiny little molecule called a microRNA, or miRNA. And as you can see in this diagram, it doesn't actually start out tiny. It begins as a long strand of RNA that gets chopped up and processed by different enzymes. It's almost like a sculptor chipping away at a block of stone until they have the final perfect shape. And that final product is a tiny, really powerful little regulator on a mission. And this is where it gets really, really clever. That little microRNA gets loaded into this big protein machine called RISC. RISC basically uses the microRNA as a guide. Think of it like a heat-seeking missile system to hunt down its matching messenger RNA target. And once it finds it, it can either just block it from being read, or it can mark it for complete destruction.
Now, there are a couple of different types of these small RNA regulators. You have microRNAs, which come from our own genes. They usually bind imperfectly to their target, which just kind of gums up the works and blocks the protein from being made. Then you have siRNAs, short interfering RNAs, which can come from outside sources, like a virus. They bind perfectly to their target, and that's a signal for the RISC machine to just slice and dice the messenger RNA, completely destroying it. So 2 different tools for some very precise gene silencing. So let's recap. We've controlled access to the DNA, we've intercepted the RNA message. You'd think that would be enough, right? Nope. The cell has one more trick up its sleeve. It can control things at the absolute last possible second, right there on the protein assembly line. This is what we call translational regulation. The messenger RNA has arrived at the ribosome, the cell's protein factory. Everything is set to go. But even at this super late stage, the cell can just hit the pause button and prevent that protein from ever existing. It is the ultimate form of real-time, fine-tuned control. Let's look at one of the most beautiful and elegant systems in all of biology, how our cells manage iron levels. Now look at these 2 diagrams. What is just mind-blowing is that the cell uses the exact same switch to control two completely different genes. And by just placing that switch in a different location on the messenger RNA, it gets the total opposite result. Let me break down how this works. First up is ferritin. This is the protein that stores iron. So, when your iron levels are low, a protein latches onto the very start of the ferritin message. This physically blocks the factory from even starting to read it. And the result, no storage protein gets made. Which makes sense, right? You don't need to build containers to store iron you don't have. But when iron is high, that protein lets go, and the factory fires up to make ferritin to store all that extra iron. Now, let's look at the flip side, the transferrin receptor. This is what brings iron into the cell. Here, that very same protein switch binds to the end of the message. When iron is low, the protein acts like a shield, protecting the message from getting destroyed. This lets the cell make lots and lots of these receptors to grab all the iron it can. Then, when iron is high, the protein lets go, the message gets degraded, and the cell stops importing more. It's just beautiful. Same switch, different location, perfect control. So we've seen these incredible little molecular machines doing their jobs, but why does any of this matter to you in your day-to-day life? Because this entire unbelievably complex system doesn't operate in a vacuum. It's constantly listening and responding to the world around you and inside of you. This slide really brings it all home. Everything we've talked about, from how your chromosomes are wound up to all those little chemical tags like methyl groups, this entire system is being influenced by all sorts of factors. And these epigenetic changes can have very, very real consequences for our health, affecting everything from our risk for certain diseases to how we age. And what are these factors? Well, they're things we all encounter every single day. The food we eat, the simple process of getting older, chemicals in our environment, even things that happened way back when we were developing. All of these things can send signals that tell those writers and erasers to get to work, changing the notes written on our DNA.
So if there's one thing to take away from all this, it's this. For a long time, we thought of DNA as this fixed, static blueprint for life. But we know now that's not right. It's much more like a dynamic script. It's a living document that your cells and your environment are constantly editing and annotating in real time. And that leaves us with a really powerful and kind of amazing question. If our environment, our choices, can actually influence how our genetic script is read, what does that mean for us? It suggests that we might just have a bigger role in directing our own cellular story than we ever thought was possible.